A very good afternoon to all of you, my dear children. Last time we have seen introduction of heredity and evolution. Okay, so in our previous class we were studying about heredity, and we have seen the introduction of heredity, whatever the discoveries made in the field of heredity we have all seen. Okay, now let's continue further. In the previous class we have seen in the year 1961, Jacob and Monod, they have discovered the process of protein synthesis. They have discovered the process of protein synthesis in bacterial cells. In bacterial cells. Okay, so till here we have seen in our last video. Now, as we were studying about heredity, why we are studying heredity, why it is important to study the heredity, क्या ज़रूरत है हेरिडिटी पढ़ने की. ओके, द थिंग इज़ हेरिडिटी इज़ स्टडिंग हेरिडिटी इज़ यूजफुल in the field of genetic engineering. In the field of genetic engineering, through this genetic engineering we can cure various genetic disorders. We can diagnose, we can bring about treatment of various genetic disorders. In the last year, in your 9th standard, you have seen various genetic disorders like sickle cell anemia, thalassemia, Down syndrome. All these we have seen about different genetic disorders. So we can study about these genetic disorders. We can prevent these genetic disorders. That's the reason we are studying heredity. Apart from that, we can make variety of hybrid varieties of plants and hybrid transgenic varieties of animals. Okay, all these we can and different antibiotics we can synthesize by using heredity in the field of in the field of microorganisms, in the field of microbiology we can say. Okay, so that is the reason heredity is studying heredity is very, very important. In your 10th standard, we will be studying important process in this chapter, that is nothing but transcription, translation, and translocation. In today's class and in the next class as well, we will be studying about transcription, translation and translocation. Okay, let's continue further. When we are studying about this transcription, translation and translocation, we need to be familiar with few terms like DNA and RNA. Let's have a quick revision from 9th standard. Okay, so what is this DNA and RNA and what are their structures? Okay, so if you have a thorough understanding about these two things, you will be able to understand transcription, translation and translocation very, very easily. Let's continue with the DNA. Okay, so DNA was discovered by Watson and Crick. We know it was a double helical structure. So we have seen in our previous class the DNA is present as genetic material in most of the living organisms. Viruses were exception. Okay, so how this DNA looks? DNA is a double helical structure. Okay, like this double helical structure. And what is this DNA is made up of? DNA is made up of 4 nitrogenous bases. Genus bases. What are these 4 nitrogenous bases? Adenine, thymine, guanine, and cytosine. Adenine, thymine, guanine and guanine, few teachers use it. Okay, and cytosine. So by using these 4 nitrogenous bases, a DNA strand is made up of. Both the strands of DNA are made up of these 4 nitrogenous bases. They can be present adenine, guanine, cytosine, thymine, 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 adenine, guanine, cytosine, 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 guanine. Okay, this is the single strand of DNA. Let's see another strand of DNA. If here it is adenine, you have to remember adenine will always pair with thymine and guanine will always pair with cytosine. That means if on one strand of the DNA adenine is present, complementary on the another strand of the DNA complementary to adenine, always thymine is present, and this adenine and thymine are bound together by 2 hydrogen bonds, and guanine and cytosine are bind together with 3 hydrogen bonds. Okay, so complementary to the adenine it will be thymine, complementary to the guanine, here it is guanine, so it will be cytosine. If it is cytosine, it will be guanine. Thymine here adenine, adenine, adenine. If adenine is here, it will be thymine. Here cytosine. If it is cytosine, it will be guanine. If it is cytosine again, it will be guanine, guanine, and here opposite to the guanine it will be cytosine. This is how the structure of DNA resembles. Now as I told you, adenine is combined to the thymine by 2 pairs of hydrogen bonds and gone into the cytosine by 3 pairs of hydrogen. Here it is 3 pair of hydrogen, 2 pairs of hydrogen bonds, again 2 pairs, 2 pairs, 2 pairs of hydrogen bond. Here guanine and cytosine has 3 pairs of hydrogen bond. Here it is 3 pairs of hydrogen bonds, and here it is 3 pairs of hydrogen bonds. So when you look at the structure of the DNA, it will resemble like a ladder. So here you have to understand adenine will always combine with the thymine and guanine will always pair, not combine but will always pair with the cytosine. Adenine will always pair with the thymine and guanine will always pair with the cytosine. If it is one strand of the DNA, the other strand of the DNA will be complementary to the base pairs on one strand. Like thymine, if it is present, it will always pair with adenine. Cytosine will always pair with the guanine. So this is a basic DNA structure as of now we are seeing, as of now, which is required to study transcription, translation and translocation. Okay, now the RNA also has the same structure. Okay, RNA also has the same double helical structure, but okay, let's see now here. This RNA is also made up of 4 nitrogenous bases, but the thing is the thymine is replaced by uracil. There is another nitrogenous base termed as uracil. In the structure of RNA, the thymine is absent and it is replaced by a nitrogenous base termed as uracil. Now in the structure of RNA, adenine will always pair with uracil and guanine will always pair with cytosine. So if we look at the structure of RNA, okay, here we'll get uracil. Wherever adenine is present, instead of thymine we'll be having uracil here. Okay, that is the major difference in the structure of DNA and RNA. DNA is also made up of 4 nitrogenous bases. RNA is also made up of 4 nitrogenous bases. DNA has the double helical structure. RNA also has the double helical structure. Here in the DNA, 4 nitrogenous bases are present: adenine, cytosine, guanine and thymine. Whereas in RNA, 4 nitrogenous bases are present: adenine, uracil, guanine and cytosine. That means thymine, which is present in the DNA, is replaced by uracil. Okay, now one more basic introduction. Now when we are talking about the DNA, there are 3 types of DNA present based upon their location. Okay, what are the 3 types of DNA? One is mRNA, okay, another is tRNA, and the third one is rRNA. Okay, these are the basic 3 types of RNA which are mentioned as per your 10th syllabus in your textbook. Now when we are talking about this RNA, it is of 3 types which you have to remember to understand translation in a better way, transcription, translation and translocation in a better way. Now RNA, there are of 3 types: mRNA, tRNA, rRNA. M stands for messenger RNA. Messenger RNA, that means which is going to carry some message. Okay, mRNA is for messenger RNA. tRNA is T for transfer RNA, M for messenger RNA, T for transfer RNA, and R for ribosomal RNA. Now, the full form of DNA is deoxyribonucleic acid. The full form of RNA is ribonucleic acid. And now, this RNA, as per your 10th textbook, it is of 3 types: mRNA, tRNA, rRNA. M for messenger RNA, T for transfer RNA, R for ribosomal RNA. This is the basic introduction and to study, so that you should, when I am explaining about transcription, translation and translocation, this is the basic introduction you should know about the DNA and RNA.